Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we will discuss control of enzyme synthesis and its pattern in different diseases. The key concepts are: what is control of enzyme synthesis? What are constitutive enzymes? What are adaptive enzymes? And what are the enzymes pattern in different diseases? Number first is control of enzyme synthesis. As the sentence indicates that enzymes are synthesized in the body to a very controlled level. The synthesis of enzyme is not the haphazard or the random process, but the synthesis of all types of the enzymes. They are under the strict control, but no doubt these like levels or enzyme synthesis may be affected to a very greater extent when body is infected with certain diseases, and therefore most of the enzymes, particularly the rate limiting ones, as the name indicates that many reactions are in many reactions there are rate limiting reactants. Same is the rate limiting enzymes. And therefore these rate limiting enzymes, they are present in very low concentration, and nevertheless the amount of enzymes directly control the velocity of the reaction which is catalyzed by that enzyme. It means that you have seen that there was direct relationship between the amount or the concentration of enzyme and the speed or the velocity of reaction which they were monitoring or undergoing. Like with the rise of the substrate concentration, yes, up to certain limits the rate of reaction rises, just like the amount of enzymes also directly controls the velocity of the reaction which that enzyme is catalyzing. Therefore, many rate limiting enzymes have a short half-life. Half-life predicts that for how much time the enzyme will like exist. Therefore, half-life of different enzyme is different. Therefore, you can say that the half-life of enzyme is responsible for investigating the working of the enzyme. And there are two types of half-life, like dynamic and static. And it means that these two half-life are no doubt measured and compared with one another. And therefore, these half-lives period, they help in the efficient regulation of the enzymes level, because I have told you earlier that enzymes are not the hazard or there's not the such type of the substances in the body which can go on the synthesis for a long period of time without any need, but there is a very controlled, you can say, dynamic relationship between the enzyme need and its synthesis. Therefore, they pass through the or they undergo the very efficient regulation system. And there are two types of enzymes. One is the constitutive, and other are the adaptive. Number first is constitutive enzymes. It means that they are the constitutive, like structure building. They are the housekeeping enzyme. It means that the level of which are not controlled and they remain fairly constant. It means that the level or the concentration of these enzymes, they remain almost fairly constant throughout the investigated period, or you can say some certain time span. Therefore, there is no need to control the levels of such type of the enzymes, which are the constitutive in nature. And second are the adaptive enzymes, and their concentration, they may rise or decrease as per body needs. It means that one body need is more certain type of the enzymes, they will be like synthesized in higher concentration, and therefore they are well regulated, and therefore the regulation mechanism is there to keep their concentration at certain optimum level. And the synthesis of enzymes, which are no doubt the proteins, they are regulated by the specific type of genes. It means that overall, for constitutive enzyme, their concentration or the levels remains fairly constant throughout or through the definite time period. But for the adaptive enzyme, their concentration may be rise, their concentration may be increase or the decrease. Number next is induction and repression, and they are derived from the well-known words like induct, induce, repress. Induce mean to make the part of that system. It means that something are coming into the system, while the repression means repress towards the decrement, decreasing, suppressing. And therefore the term induction is used to represent increased synthesis of enzyme, while the repression indicates decreased synthesis of enzyme. That induction means yes, in the body or in the certain cell or the tissue, the level is like a fall. Therefore, it should be made at certain optimum level, acceptable level. Therefore, the enzyme must be synthesized into that cell, mitochondria, tissue, livers, and so on. And therefore, induction means the increased synthesis of enzyme, whereas the repression is the reverse one, like it indicates a decreased synthesis, that body no longer need this enzyme. And induction or repression, which ultimately determine the enzyme concentration at gene level through the mediation process. And this mediation means resolution, resolving certain problem, process, or help out. And this help or resolution is provided with the help of the hormones or certain other substances which are present in the body. It means that this induction and repression is the well regulated system, and it's responsible for controlling the enzyme concentration. And it's through the you can say it's by the gene is responsible for controlling them, and it is mediated or help out by the involvement of hormones. Number next is example of the enzymes induction. Hormones like insulin, it induces the synthesis of glycogen synthetase, glucokinase, phosphofructokinase, and the pyruvate kinase. It means that insulin, which is the hormone, it will introduce or it will responsible for the increase in the synthesis of certain enzymes, and these enzymes are like glycogen synthetase and glucokinase and pyruvate kinase and so on. And you have studied all these enzymes in the metabolism of the glucose, metabolism of glycogen, and they all are involved in the utilization of the glucose. It means that insulin is also responsible for the utilizing these glucose molecules in the body. But this insulin hormone, it will show its function through the synthesis of these enzymes. And it means that all these enzymes are involved in the breakdown of the glucose for providing the energy. And therefore the hormone cortisol, it induces the synthesis of many enzymes like pyruvate carboxylase, tryptophan oxygenase, and tyrosine amino transferase. Next is the example of repression. Repression means that yes, there will be less synthesis of certain enzyme, or the synthesis of enzyme will be repressed. It should be suppressed. It should be stopped. In many instances, like the substrate, they can repress the synthesis of enzymes. Enzyme is pyruvate carboxylase, and it's a key enzyme in the synthesis of glucose. And you have seen it when we have discussed the gluconeogenesis, which was the synthesis of glucose from the non-carbohydrate source, which may be pyruvate, which may be certain amino acids, and therefore pyruvate carboxylase, it's an important or the key enzyme for the synthesis of glucose when the non-carbohydrate sources are available. And therefore, if there is a sufficient glucose available already in a tissue, in a cell, and then there is no necessity for the synthesis of glucose, and therefore the glucose synthesis will be stopped, will be repressed, will be suppressed, and it will be achieved through the repression of enzyme, which is the pyruvate carboxylase. It means that when pyruvate carboxylase will not be present, then you can say there will no need to for the formation of the glucose synthesis, and it this enzyme is repressed by the glucose units itself. It means that this stopping of the glucose synthesis is achieved by the repression of the carboxylase enzyme, and this repression is done by the glucose which is already present in the cell. It means that it is the example of the enzyme pyruvate carboxylase repression. Next is enzyme degradation. Degradation means breakdown of enzyme, and enzymes are not the immortal. Immortal, they are not the such type of the species which can't be degraded, which can't be breakdown, but they are the mortal one. They can be go towards the end, since it will create a series of problems if enzyme were mortal. Therefore, enzymes are not immortal species. They can be degraded, they can be breakdown, and immortal, you can use this terminology like for eternal life, for non-ending life. Therefore, the such type of the biological moieties they are not immortal. And there is lot of variability in the half lives of individual enzymes. For example, it is in days, while in while for others it may be in hours or in minutes. It means that the half-life period for certain enzyme, it may be break down or degrade in few days. It may degrade in few hours, or some enzyme can degrade in few minutes. It means that yes, they are synthesized, they are utilized, or if they are not utilized, they are degraded, they are break down in few minutes. Like LDH4, it is its half life is like 5 to 6 days. LDH1, it is 8 to 12 hours only. And for amylase, it's 3 to 5 hours only. It means that these enzymes are going towards breakdown. And in general, the key and the regulatory enzymes are most rapidly degraded. It means that the important enzymes which are key to certain metabolic process, like we have discussed the carboxylase enzyme, and the regulatory enzymes, they are more rapidly break down, and if not needed, they immediately disappear. Like they are synthesized, they are present in the cell, but their need is not felt by the cell. Therefore, these enzymes will be disappear or break down or degraded immediately. And when they are required, when the cells feel their need, and they are quickly then synthesized. And though not always true, an enzyme with long half life, they are usually sluggish one. They are very slow one. It means they are not the active one in their catalytic activity. Next is the enzyme pattern in different diseases. Pattern mean how the enzyme concentration varies, how they are responsible for the onset of certain diseases, which may be associated with like heart, with liver, with muscles. And for the right diagnosis of a particular disease, it is always better to estimate a few, three or more serum enzyme instead of a single enzyme. It means that when you will collect the data for the estimation of three or more enzymes, you will in fact gather much information for the diagnosis of certain important body organs like kidneys, like liver, heart. No doubt these are the very important organs. And there, for example, of the enzyme pattern in different or the important diseases, they are given over here. Number first is enzyme in myocardial infarction, which is associated with the um, some problem in the, you can say, heart muscles, and therefore the enzymes, namely creatine phosphokinase, which is called as like CPK, aspartate transaminase AST, and lactate dehydrogenase like LDH. And it means that these three, like CPK, AST, and LDH, they are important for the diagnosis of the myocardial infection. It means that three types of the enzymes will be investigated in the diagnostic lab in order to collect information about the proper functioning or the proper condition of the heart muscles. And therefore, the elevation of these enzymes like CPK, AST, and LDH in the serum in relation to hours or the days of the myocardial infection, it's important in its diagnosis. It means that blood sample will be taken after few hours, after some days, and investigation will be done at which level or to which extent the concentration of these enzymes has been elevated in the blood of individual which has the problem of some heart attack or heart pain or chest pain and so on. And next is the creatine phosphokinase, which is precisely called as the isoenzyme. And in last lecture, we have also discussed that its isoenzyme like MB is important, although it is present in combination of like BB, double M, and MP, but MB is important in this case. And it is the first enzyme to be released into circulation within 6 to 18 hours after the infection. It means that if the person has the problem of myocardial infection, then it's the first enzyme which will be considered in the which will be seen in the blood of the that individual or the patient, and only after the within the 6 to 18 hours when this problem will be raised. And therefore CPK, like creatine phosphokinase estimation, it's highly useful for the early diagnosis of myocardial infarction MI. And this enzyme reaches a peak value within 24 to 30 hours, like uh, within a day, about when the person will be suffered with such type of the heart problem, and returns to the normal level by the second or the third day, where when its level will be going towards normal, when collapsing or this situation will going to be like normalized. And the second is the aspartate transaminase, which is AST or SCOT, and it rises sharply after CPK. Like first CPK will be raised, and after CPK, this AST will be raised, and it reaches within like 48 hours of the myocardial infection, and AST takes nearly 4 to 5 days to return to its normal level. Next is lactate dehydrogenase LDH1, and generally we have seen that this rises from the second day after the infection. It means that we have investigated all these enzymes started from here CPK, AST, LDH. First rise was CPK. After that, AST, and lastly, it will be the LDH, which rise and from the second day after the infection. And about it will take 10 to 15 days to reach its level. It means that it will take long time, and thus LDH is the last enzyme which will rise. It will show high level, and therefore the last enzyme to return to the normal level in the myocardial infection person. And next are the cardiac troponins, troponins like the CT. Although it's not the enzyme like LDH, CPK, it's the protein. And the proteins, cardiac troponins, they are highly useful for the early diagnosis of the MI level or the MI, like myocardial infarction situation. Among these troponin, one which is it's the inhibitory element of the actinomycin adipase, and troponin T, and these like troponin I and troponin T, they are important. And cardiac troponin, it's released into circulation within 4 hours after the onset of myocardial infarction. and thus reaches a peak value by 12 to 24 hours, it means within a day, and it remains elevated for about a week. It means that it also takes a long time to return to its normal level, like that of LDH, which nearly takes like 2 weeks to return to its normal level. And here is the representation of the person with a condition of heart attack. And you can see that there is less blood supply to the heart, and there is blockage in the artery, and the muscles are also badly damaged. They are not the healthy muscles, they are broken muscles, damaged muscles. Next is the enzymes in the liver. It means that enzymes which are responsible for the cardiac function. Yes, they are important, and they are not the single enzyme, but they are the multiple enzymes which rises at the different levels, which rises at the different time period, and therefore they leads toward the onset of and they represent how the heart is going to be damaged. The heart muscles are affected with the heart attack condition. Therefore, next is the enzyme in the liver diseases. It means that there are many enzymes which are like their levels have been raised, their levels have been changed in the uh, disease liver condition. And following are the enzymes, which one elevated in the serum. They are useful for the diagnosis of the liver dysfunction. And liver dysfunction, which is due to the viral hepatitis like jaundice, it may be due to the toxic hepatitis, cirrhosis, or the hepatic necrosis. And we have discussed the back terms already in a very well way and in detailed way. Only remaining is the hepatic necrosis. And what will happens in the hepatic necrosis? It resembles like an acute or the toxic injury to the liver, which may be sudden, and it's responsible for the marked elevation in certain aminotransferase enzymes. And for it, hepatic necrosis, the early signs of like hepatic or the is the dysfunction or the failure, and there are many reasons that causes this hepatic necrosis. It may be due to the drug toxicity, or it may be due to the certain Wilson disease and so on, which is also responsible for this type of the hepatic necrosis problem. And enzymes which are important, they are the alanine transaminase, aspartate transaminase, lactate, dehydrogenase, and enzymes that markedly increase in the intrahepatic and the extrahepatic cholecystitis are the like alkaline phosphatase. It's a nucleotidase serum, glutamyl trans peptidase, and it's useful in the diagnosis of the alcoholic liver disease. And next are the enzymes in the muscle disease. Which enzymes are important? In the muscular dystrophy, like Like we have discussed in the last lecture, what is the muscular dystrophy, which is stands for MD, and it's a group of muscle diseases, and that result in the increasing weaknesses, which is going to build up in the muscle. Muscle will be going to be weak day by day, and the breakdown of skeletal muscles uh, will go on with the passage of time, and the serum level of certain muscle enzymes are also increased, and these include creatine phosphokinase, aldolase and aspartate, transaminase. Like it's the condition of muscular dystrophy, and in this condition you can see that the normal muscle is well healthy one. It's uh, uh, it's thicker in size, but with the passage of time, the person with such type of the disease, the muscle will going to be weak, and therefore muscular strength will be going to be decreased with the passage of time. It's all about today's lecture. And for assignment, here are questions like what is the genetic induction, what is feedback repression, and lastly what is difference between an inducer and the repressor. Best wishes, and Allah Hafiz students.